Hello students, welcome back to the channel. In this video, we are going to study what are the important things which play a role in the second line of defense. See, first of all, immune system, it is innate and acquired. Innate is the one which is non-specific. It is first line of defense, which is actually made by the skin, mucous membranes and certain chemicals. But if suppose pathogen is successful in gaining the entry, you can see if it is successful in gaining the entry inside the body, then the second line of defense come. Now, second line of defense, what are the cells which are included in the second line of defense? Means let us study what are the cell component of the second line. First is phagocytes. The cells which are phagocytic in nature, they are mainly neutrophils and macrophage. But dendritic cell also do the phagocytic nature. Okay, so what they're doing it to communicate to each other, these phagocytes will release a substance called cytokines. And they are important cell signaling molecules by which they can protect against pathogen and guide the immune cell to the site of infection so that phagocytosis can be done easily. Natural killer cell. Next important component is natural killer cell. They will target abnormal cell and virus infected cell. Then is mast cell. Mast cell is the one which will release histamine, which is actually vasodilator, and it plays very important role in the inflammatory response. Next important cellular component is eosinophil, which is actually releasing DNAs, RNAs, protease to destroy these components of the pathogen. So basically the cell component which play very important role in the second line of defense are your phagocytes, natural killer cells, mast cell, and eosinophil. Understood, Peter? These are cell component. Let me show you through one diagram. See, we already studied this diagram. Okay. We studied this diagram. In the previous video, you can check it, hematopoietic stem cells. So see, these are your neutrophils, macrophage, and dendritic cell. They are phagocytic cell. Natural killer cell is not phagocytic. It will do the programmed cell death, virus infected or abnormal cell. Please check the video for this also. I've made it separately. Then mast cells, they release histamine and plays very important role in inflammation. And eosinophil also release certain enzymes to digest the biochemical component of the pathogen. Now, non-cellular components. Non-cellular components of second line of defense. First is cytokine, complement proteins, inflammatory response, and fever. So see, beta, what are cytokines? They are actually soluble proteins. They act as a communication signals. Is it clear? And these cytokines, they can function depending upon that from where they are released and where they target. For example, if you see here, the chemical which is released from one cell is having receptor on its own surface. So this is autocrine type of signaling. Means they are stimulating their own cell only to produce more and more cytokine. Now, sometime what happened, the secretory cell is having the receptor nearby only. Nearby cell is having the receptor. Very short distance they have to travel. They can go by diffusion also. So that is paracrine signaling. And if suppose the chemical are moving through blood vessel to the distant locating cells, means the cell which is very far from the secretory cells, then we say endocrine signalings. So these cytokines, they will be released to stimulate production of some other mediators so that the cell division, cell proliferation, or inhibition of cell division, many things they can work. Clear, beta? Now, important classes of cytokines. Cytokines has important classes. One is interleukin, which is actually secreted mainly by macrophage and lymphocytes, and they regulate interaction between WBCs. Clear? Then is chemokines. Chemokines actually they are chemotactic in nature. And their work is to recruit lymphocytes or leukocytes at the site of infection, means during inflammation. Then is tumor necrosis factor. It is secreted mainly by the macrophage and by T cells, and their work is to control the tumor spreading. They will destroy the tumor cells. And interferons, it is actually immune signaling molecules, very important against virus infection. So these are the important classes of cytokines. Cytokines are chemical proteins which are released from one cell and give the signal to the another cell. So I think so this much is easy for you. Now see next. Types of interferon. Type 1 will include alpha and beta. And if you see here, this is the virus, which is cell which is infected by the virus. So what happened? This gene is now making protein. Interferon is synthesized and it is released, and receptors are present nearby, neighboring cell. Clear, neighboring cell receptors are present. Now interferon will bind to the receptor of the neighboring cells, and now they will produce antiviral protein to block viral replication if suppose cell become infected with the virus. So antiviral protein genes are made so as to prevent viral infection, or if suppose infection is done, to prevent replication, and they are targeting neighboring cell. So which type of signaling is it? It is paracrine signaling. And this is special type of cytokine which is released, it is a interferon, especially effective against virus infected cell. Now next is type 2. This type 2 is actually very important in the activator of immune cell. It is the one here we are trying to activate our immune system so that immune system identify the abnormal cell, cancer cell, abnormal cell as foreign and destroy them. Is it clear? So we are in this type macrophage, they stimulate macrophage to destroy tumor cell. Okay, type 2 interferon will stimulate macrophage to destroy tumor cell. So I hope so up till here concept might be getting easy better. Now see next, complement proteins. Okay, this is also non-cellular method of second line of defense. Is it clear? Non-cellular method. 30 types of proteins are present in the blood plasma, which are present in inactive form, made by the liver and present in blood plasma. Now, these complement proteins, they are actually represented by the symbol C. You are writing the number. Obviously, number is there. Many types are there. So we are writing 5, 6. You can see here, these are the numbers we are giving it. And sometime there is A and B type also. So we are writing it after that small letter also. And they will start a one cascade reaction, one or another, so that reaction result can be amplified. Now see what they will do. Either they will do opsonization. Complement protein will stick to the outside of the surface of pathogen, make it easier for the cell of immune system, means for the phagocyte to identify as a foreign and then destroy it. So opsonization of pathogen is done by the complement protein so that phagocytosis becomes easy. Then second is chemotaxis. They gather near the pathogen and attract phagocytes. So complement proteins are gathering near the pathogen and attracting the phagocytes. Then is lysis. They will make on the surface of pathogen one membrane attack complex. This complex they will make so that the pore can be created and water enter and the cell go for lysis. Previous slide you can check here also. This is the pore which is made by the membrane attack complex so that the water enter and lysis can occur. So I think so in non-cellular we have covered till now two things. One is cytokine, another is complement. Clear, beta? In cytokine we discussed interleukin, then we discussed TNF, we discussed about interferon, type 1, type 2 also, and now we have discussed complement proteins. Next is inflammatory response. So inflammatory response is 4 responses are there: redness, swelling, heat, and pain. Is it clear? It is a response to the infection or to the injury. Now see here, 3 steps are there. First is vasodilation. For vasodilation, mast cell will release histamine. Macrophage will release cytokine. And impact is this, ki the blood vessel will become dilate. When the blood vessel is dilating, more blood flow is coming, so it warms the skin. And because blood is too much coming here, so redness is causing there. Now permeability increases. Histamine and other compounds, they will increase the permeability. So fluid and antibodies will come out. Is it clear? They are coming out. Now volume of interstitial fluid is increasing, so swelling is going on. Now enzymes in the plasma, they are causing the pain. That is a characteristic of the inflammation. Now because of the increased blood flow, large number of neutrophils and other phagocytic cells, they are coming and then they will do the phagocytosis. So phagocytosis is going on, and as a result, this is the slowly what will happen. This inflammation will be covered, and growth factor from white blood cells and platelet will stimulate cell division and make the healing. So this is the way healing is done. Clear? Next is fever. Fever is an inflammatory response which extends beyond the site of infection and affect the entire body. The overall increase in the body temperature. You know one thing, normally body temperature is maintained by hypothalamus. But certain bacterial or viral infection can release, means you can say that some chemicals are released. That chemicals are known as pyrogen, which will affect the hypothalamus to change its thermostat, means it will change the setting. For example, if you check here, one LPS, this is a toxin which is produced by gram-negative bacteria. It is an example of exogenous pyrogen. Now, this exogenous pyrogen will induce the leukocyte to release endogenous pyrogen. For example, interleukin 1, okay, gamma interferon, tumor necrosis factor, all this will be going on. And as a result, what happened? The hypothalamus will reset to increase the temperature and the person will get fever. Clear? So I hope so in this short video, what are the topics we have covered? Let me just revise fast. Basically, we have to understand what are the cell component for second line of defense. These are the 4 cell component for second line of defense. Phagocyte, please learn 3 types of phagocytes are there. Clear, beta? Then go to the non-cellular component. Cytokines. Cytokines, how many types we discussed? We discussed 4 types of cytokines. Then complement proteins, inflammatory response, and fever. For cytokines, see here. First is interleukin, then chemokines, tumor necrosis factor, and interferon. Clear, beta? Interferon, alpha and beta, which is against virus infected cell. Second type is gamma, which is actual activator of immune cells. Clear? Cytokines types and interferon types. Now complement protein, total 30. And they are responsible for cascade effect. That cascade effect can be opsonization, can be chemotaxis, or can be lysis. Then inflammation. Inflammation will be having the 3 response. First, vasodilation, then blood capillarity will increase and phagocytosis will be done. So 4 symptoms will come, that is warmness, redness, swelling, and pain. And last is fever. Fever is because of chemicals which is released by the virus or bacteria infection. That chemical is known as pyrogen, which will activate, which will change, will make the hypothalamus to reset the temperature. Is it clear? So as to initiate fever. So I hope so this short video might be helpful for you. Thanks for watching. And if you like this video, please subscribe my channel so that I can be motivated to make more videos.